I guess I'd like to thank you for coming. It is my pleasure to introduce to you Samia Kurien. She hails from, that's where you have to hail from. She is an, oh, I don't know how to say it, Ethio-Indian lady. And we have been very privileged to have her at NNU for these years. She's been in my lab for the last 2 years, and she did research before that with Dr. Strohmeyer. So she's got a broad range of experiences in this. She's going to spend, we hope, the next year in the area, and then we hope to send her off to PA school so that she can be Dr. Ethio-Indian lady, as it were. But no, really proud of Samia and what she's done, and looking forward to her getting to explain to you her project on beta-3 alcohol dehydrogenase.
Thank you, Dr. Chase, for the introduction. I had the privilege of working with the kinetics of human beta-3 alcohol dehydrogenase.
Oh, it's not working.
Yeah, as I was saying, we had a problem after when the batteries changed, it wouldn't.
Alright, so genes affect susceptibility to diseases. Take, for instance, Angelina Jolie. Just last year, she learned that she carried a faulty copy of the BRCA1 gene that made her more likely to develop cancer. And so this led her to get a mastectomy. Today, I'll be talking to you about a gene that is believed to reduce the likelihood of fetal alcohol syndrome and alcoholism. I'll give you the background of alcohol dehydrogenase, and I will share with you the hypotheses that have been stated in previous studies. I will also go over the purification methods and the enzyme assays that we carried out in our lab, and I will share with you the results that we obtained from last summer. Alcoholism affects 15 million people in the United States. High consumption of alcohol in mothers is known to cause severe brain damage in the developing fetus. As you can see in this image right here, this shows you a normal brain, and this is a fetal alcohol syndrome brain. It's more shrinked. And statistics have shown that about 1% of babies born in the US have fetal alcohol syndrome. Alcoholism and fetal alcohol syndrome can be affected by genes. For example, 1/4 of the African American population is protected against fetal alcohol syndrome because they carry the beta-3 ADH isoform. There are 5 classes of the ADH isoforms, and some of these isoforms, there are 5 classes of ADH, and they are known to have some isoforms. Our team worked with the beta-3 class, and they have 3 polymorphisms, namely beta-1, beta-2, and beta-3. And they are present at high levels in the liver and in the stomach. My lab partner, Joshua Benton, and I worked with the beta-3 isoform. ADH is responsible for ethanol metabolism. Ethanol and NAD are oxidized by alcohol dehydrogenase to form acetaldehyde and NADH. These substrates are important, and they should be considered especially, and they should be varied when we're trying to study the kinetic constants. The question is, how fast does beta-3 break down ethanol? And most of the researchers base the rate of the breakdown of ethanol by beta-3 by determining the Vmax. Before I explain these figures, I'm going to define 2 kinetic terms for you, which are Vmax and Km. Vmax is the maximum velocity of an enzyme when it is fully saturated with a substrate, and Km is the substrate concentration at which the enzyme is half saturated. So, due to the high Vmax of beta-3 relative to beta-1, researchers believe that mothers with beta-3 process ethanol more than mothers that carry beta-1. But these values right here do not give us enough information on how much how much concentration ethanol is needed for the enzyme to be fully saturated. So what we did is we collected kinetic constants from other literature data, and we measured the ethanol oxidation as a function of ethanol. And we found that beta 3 does indeed have a higher Vmax than beta 1. So, this graph does agree with our hypothesis, but we do not fully agree with theirs. And this is because we found that the beta ADH makes up only a small portion of the total liver ADH pool. And also, when calculating the ethanol oxidation rate as a function of ethanol and acetaldehyde, we found that beta 1 oxidizes ethanol oxidation faster than beta 3. So that doesn't really agree with the original hypothesis. And the second reason why we're not fully convinced with the hypothesis of beta 3 being oxidizing ethanol faster than beta 1 is because not much literatures have been done, not much study has been done at physiological conditions. Kinetic studies have only been done at standard temperatures. So we determined the kinetic constants for beta 3 ADH at physiological and standard conditions. We wanted to test the hypothesis of beta 3 to see if it does oxidize ethanol faster in tissues. But before we could get to that stage, we had to go through various processes. The first one being transformation. We were given recombinant beta 3 plasmid, and Dr. Steve Pekovich, he transformed the plasmid into the E. coli cells. The E. coli cells were grown and induced with zinc sulfate and IBTG so that more enzymes can be made. Then these cells were lysed so we can release the enzymes. So once the bacterial cells were lysed, we carried it through different purification processes. This shows you the series of ion exchange columns, and what happens is when we run it through the DAE, it binds to the unwanted proteins, and we collect the protein solution and put it into the separose column. And the separose column binds to the beta 3 protein, but there is still some unwanted protein that goes out, and AFI group binds with the ADH protein, but that's like the purest column that we run it through. Then once we've done those steps, we check if we do have a purified enzyme, and we run it through a gel. The gel tells us like the extent of our enzyme purity. So here's a sample of what our gel looks like. We run the samples into these gels. This is the marker length, and you can see how dense it is when it starts out, but as you keep going through the different purification process, we get a single band at the 40 kilodalton marker, and this confirms the presence of our pure beta 3 ADH. Now that we have obtained our beta 3 ADH, we're able to carry out our kinetic assays and proceed with our kinetic studies. The pure ADH assays were performed with varied concentrations of ethanol and NAD, and they were performed with varied concentrations of acetaldehyde and NADH at both standard and physiological temperatures. We measured the rate of the reaction on the spectrometer, and then we calculated the kinetic constants from the Sigma plot software, and then we fit these rates into the following equation. The following slide will show you the type of results we get. So this shows the rate of acetaldehyde at 5 different NADH concentrations, and like these little dots are triplicate measurements of the rates of the acetaldehyde, and the software just calculates the parameters of that best fit to our data. We did this at 25 degrees Celsius with the same substrate concentration, acetaldehyde and NADH. We also did the same thing with ethanol and NAD at both physiological and standard temperatures. And oh, I forgot to mention that the R square right there is high us the confidence that we did obtain good kinetic constant values. So now that we did gather all that kinetic information, we are able to compare it with the literature study and compare the results we got at the two different temperatures. And we can see that kinetic constants of beta 3 ADH are significantly affected by temperature. The Vmax is higher at 37 degrees Celsius, and the Km for ethanol and NAD is lower at 37 degrees Celsius. But now that we do have good numbers, does that mean beta 3 is faster at oxidizing ethanol? This plot right here is our results when we measured the ethanol oxidation as a function of ethanol. And it does show that beta 3 does have a higher Vmax, and this was the literature value that we used before. And this one, if anything, proves our results proves their hypothesis better, that beta 3 does oxidize ethanol faster than beta 1. But when we look at it in the liver tissue, we find that the beta 3, when we're measuring the ethanol oxidation as a function of ethanol, beta 3 is slightly higher. But when we look over here at the ethanol oxidation as a function of acetaldehyde, beta 1 still processes ethanol oxidation a bit faster, but we see a little bit of beta 3 being involved with the ethanol oxidation. So it's hard to say that beta 3 is faster at oxidizing ethanol, but with our kinetic results, we can conclude that it does support the hypothesis that beta 3 is protective because it does oxidize ethanol faster than beta 1, but not under all conditions. So to wrap it up, BRCA genes are the only genes that determine the risk of developing a disease, but beta 3 genes does affect the susceptibility to alcohol-related diseases, possibly. If we were to compare that graph, we're not really sure. But for now, we do think beta 3 does play a role with alcohol-related diseases. I'd like to acknowledge INBRI for providing the funds for NNU to allow us to continue with research. I'd like to thank Dr. Chase and Dr. Nogales for giving us the opportunity to work with you, and Joshua Benton for being a great lab partner, and Nancy and Steve for helping us a lot in the lab. I'd like to thank the NNU faculty, staff, and my friends. I could list them all, but it would be as long as my presentation. And I'd like to thank the Stelway family and the Park family for being very supportive. And last but not least, Dan Miller, who helped me come to NNU. And that sums up my presentation. Any questions?
So your results that you showed that were related back to liver oxidation, were those done with actual liver samples, or were those through a model that's trying to model the liver?
It was through a model. We just actually, the one on the right is our results. We just combined them together.
Okay.
On the left, I believe that it was from the literature values that we just combined it together. Any other questions?
Taking the jacket up. It's pretty snazzy, dude. I could never get to wear it. I would totally wear it. I was looking forward to my interview, and then I was like, what interview? Where'd you get interviews? I got Rocky University of College, Victoria University.
It's in San Francisco.
Yeah, and they got accepted that book.
No way. Dude, that's awesome.
Congratulations.